Hi everyone, Christer here from Master One Thing. Today we have the honor to have Professor Pankaj Kapahi as our guest in the podcast. Pankaj has his own lab at the Buck Institute for Research on Aging. His research for 2 decades has been focusing on things such as dietary restriction, fasting, rapamycin, nutrition, and longevity. Pankaj was also one of the pioneer researchers who discovered in the beginning of 2000 that genetic inhibition of mTOR led to life extension in mice. In today's podcast, we are going to discuss about this discovery, but also about calorie restriction, fasting, GLYLO, and lots of other interesting topics. So, Pankaj, welcome to the show.
Pleasure to be on it. Thank you for inviting me.
A good way of starting this conversation is to go back in time in the longevity field. Can you talk a little bit about how this field got started?
Yeah, I mean, I have to say my introduction to the field was I was interested in aging, but when I first read about the studies on caloric restriction, which were done in the early 1930s, that limiting calories in mice extended lifespan by over, could be even 50%. That really got me excited. And I remember when I was a teenager walking around thinking, I have this special knowledge that just eating less could give me an extra decade or 2 in life. I was very excited about this. And since then, I have tried to not overeat during my meals. I love food, but I try to pay attention to it because just, the potential for what eating less could do, we have not, I think there's a lot more to learn about this. And I think there's very interesting lessons being there for the field as well on this topic. Yeah. And it's been on 100 years, but I think there's a little less than 100 years, but there's a lot more coming in the field.
Yeah. When you came into the field, was it easy to find a position in the longevity field when you started?
Oh yeah, actually, when I was doing my PhD in England, it was tough, and I remember my high school teacher said, "Don't talk about aging at your interviews." I mean, so that was tough. But I remember when I wanted to do my postdoc with Seymour Benzer, I was very clear I wanted to study the genetic mechanisms of dietary restriction. And then a paper came out while I was researching. That paper came out from Lenny Guarente on the SIRT2, and I thought, oh, well, it's been figured, and I was wrong. And I'm glad I continued, and I got very interested in like the very simple idea that if limiting nutrients would extend lifespan, could you genetically inhibit the pathways like TOR, which was there are a lot of interesting studies emerging on TOR, how that senses protein and other nutrients as well, and that what if you genetically inhibit that, would you mimic caloric restriction, the effects of caloric restriction? And I want to say at that point, I also appreciated it was not just calories that's important for lifespan extension. The word caloric restriction is used commonly, but it should be dietary restriction. And a lot of the research we have done shows that actually just limiting protein components in the diet, or and from other people, and also limiting nucleotides, for example. So we've shown studies that limiting purines also extends lifespan. And the converse is also true. People who have gout, hyperuricemia is very common. 20% of the population has hyperuricemia. That actually limits lifespan. So it's not just calories, but you can imagine, because all these nutrients work in conjunction, the idea was that evolutionary speaking, the idea is that if you limit the nutrients, the organism goes into a sort of defense mode. What what's happening is, imagine an animal in the wild, when it goes into defense mode, what it's trying to do is waiting for the opportunity when nutrients will be abundant, and then reproduction will be essentially normal. But till then, what you're trying to do is buy time, right? And I think what we are trying to do with these sort of this adaptation to sort of buy time seems to be there from bacteria to humans potentially, and the idea is to sort of activate the sort of survival mode, defense mode, where the animal, where the organism goes into improving sort of defenses so that it can live longer.
When we have that knowledge, it makes it possible for us to start hacking those processes so that we can, for example, increase our lifespan when we go into the survival mode instead of being continually in the growth mode.
Right, absolutely. I mean, hacking is more a new term, but yes, people have been trying to, utilize things like eating less and exercise for a long time to sort of improve their health. And also, I think one of the things people fear, but it's sort of counterintuitive, is it actually helps you in the short term and the long term. So again, I like to think about this in terms of what's happening in terms of evolution. When you're eating less, and the idea is to have this right sort of window of eating less, right? Then you become more alert. So studies show that short-term dietary restriction in humans will also improve short-term memory, right? And you also become more physically active. And if you think about it, what you're doing is you're essentially preparing for the survival mode, and you're making sure that you are better off at finding food, essentially, right? So you become a little more smarter so that you can locate the food. You're more active, and we recently did a study where we showed that even eyesight improves. So, and also there's a delay in the sort of age-related decline in visual function using flies. We showed this, and it's quite a dramatic effect, that how circadian functions improve, and you actually improve these different outputs in sort of short term so that you can maximize the potential to find food. But if you just don't get the food, keep the animal in this sort of hunger state, then you can sort of reap the benefits of dietary restriction.
Yeah. If we look at calorie restriction and dietary restriction, what is the difference between those two?
Yeah, so initially, a lot of studies done by Ed Mazzaro on caloric restriction in mice showed that perhaps limiting calories was the key component to to maximizing lifespan. But now, because dietary restriction has been done in over 30 different species, we are learning a lot more. So, for example, in Drosophila, they're fruit flies. They subsist on fruit. Limiting yeast seems to be critical, and yeast is their main source of protein and lipids. And what's interesting is we can get very dramatic extension in lifespan, 40-50% or more, just by limiting the yeast component, even though the carbohydrates are the same. Now, so people worry about what does that mean in terms of humans? I think a lot remains to be understood, but the same is also true in mice. Just methionine restriction or tryptophan restriction is sufficient to extend lifespan. So I think there's this sort of wave of people trying to eat more protein to build muscle mass. And surprisingly, what we have found, actually, it's there in multiple species, limiting calories does not seem to it seems to spare the muscle, right? So, and I think, again, it's worth thinking about what's happening under caloric restriction. And I want to say not going too far, right? So if you go too far, it could lead to muscle wasting, but there's a sweet spot where you actually become more active. So what we found in flies, surprisingly, is that the animals become way more active. And this means that the muscle is more engaged, actually. So we see improvement in sort of a lot of increase in the muscle protein synthesis. And it makes a lot of sense because, the muscle is a key part that if you're using that under caloric restriction, then that's going to be beneficial, right? Or dietary restriction. So, and we've also done an interesting experiment where we showed that, dietary restriction works even better when you're more active, essentially. So if you reduce the activity of flies, right, then you don't get the maximal benefits of dietary restriction. So we think, you need, in addition to limiting food, you also need the behavioral change of actually being more active to get more out of dietary restriction.
Yeah, very, very interesting. Is there some kind of biomarker or some kind of sign that you can look for so that you don't overdo things?
That's a great question. No, I'm unfortunately, I'm not sure we have biomarkers yet. And one of the things I want to say, that's kind of interesting question. You said it varies across people. I think one of the key things I want to get across in today's interview is that inter-individual variability has a huge role to play in our responses to diet restriction, rapamycin, and any drug, pretty much. That's why a lot of clinical trials, we need to sort of embrace this precision medicine approach. But the same needs to be also thought in terms of protein restriction or dietary restriction and exercise, because all these things don't work equally for everyone, and the amount of dietary needs might be different between people, between sexes, and at different ages. So I think we need to, we're not there. A lot of research needs to be there done to understand the biomarkers, like what you're asking, which ones to optimize. But I think it's important to kind of remember, the figures are sort of interesting. More than 50% of Americans overeat protein compared to what the recommended daily allowance is. And one of the flip side is we don't want to take in too little protein or even carbohydrates. Like complex carbohydrates are very good for you, but when you totally go on a sort of keto diet without carbohydrates, could be also detrimental. So we need to be careful about going too far on these, but also too much protein, especially if you're not exercising, can be damaging, and because it puts a lot of load on the body, and the metabolic products that are made, like especially things like ammonia, they end up being toxic to cells. And we're doing an interesting study where we're finding, especially in models of Alzheimer's and frontotemporal dementia, tauopathy, high protein is very detrimental to the animal.
If we go a little bit back to the discovery you did in the beginning of 2000 regarding the genetic inhibition of mTOR, can you talk a little bit about that?
Yeah, that was a fun time, and I have to say a lot of the reasons, like I mentioned, came from this sort of thinking about aging in terms of evolutionary biology, and sort of realizing that putting the animal in survival mode improves the defense responses, and that worked out beautifully for inhibition of TOR. And since then we've done quite a few other studies sort of understanding the mechanism as well. So we found that not only TOR but also the downstream components, if you inhibit them, and most interesting, protein translation. So inhibition of protein synthesis, like through S6 kinase or overactivation of 4-EBP, we found that actually extends lifespan. So this was sort of a paradox again, because it's known that protein synthesis declines with age, right? So why is it then lowering protein synthesis would actually extend lifespan? So we found this in both flies and worms, and one of the most interesting things that we found was protein synthesis. Overall reduction in protein synthesis, same thing with dietary restriction, does not mean all proteins are just going to go down. So what's very interesting is the organism sort of reprograms to decide, okay, what's more important and what should it maintain. So we found, for example, when you inhibit TOR, you actually increase the translation of nuclear encoded mitochondrial genes. So it actually improves mitochondrial function. Then we also found that the reason this is happening is the animal is burning more fat and utilizing, instead of using sugars or instead of being reliant on glucose metabolism, it's actually more reliant on fat metabolism. So you need improvement in mitochondrial function, oxidative phosphorylation, and fat recycling is critical, right? And then of course autophagy becomes critical, and all these things. So there are multiple downstream events that sort of now being characterized to sort of understand how TOR mediates lifespan extension. And another interesting one is sort of enhancement of circadian functions. So the what we call the amplitude of circadian rhythms sort of improves on dietary restriction, and we've shown that this is important for the eye and also for the fat turnover that takes place on dietary restriction. And some of these are regulated by TOR as well. Yeah. So I mean, and the other idea, and sort of just going back to the protein synthesis part, is lowering protein synthesis also ensures better quality proteins essentially. That's one of the ideas that's that's why it may be improving essentially cellular homeostasis and allowing lifespan extension.
Yeah. If I understand it correctly, the protein restriction also leads to lowering TOR activity.
Right, correct. Yeah. In flies, we found that inhibition of TOR sort of mimicked the effects of dietary restriction. But I have to say, it's not been as simple in mice. That has not panned out to be true, and it remains to be seen what happens in different systems. But first of all, I want to say there are multiple ways to sort of, like I said, if you think about dietary restriction, there are multiple mechanisms at play. It could be sugar restriction, it could be protein restriction, it could be also purine restriction and intermittent fasting. Different mechanisms. Some common mechanisms are at play, and then some may be different. So I think it remains to be seen in more other systems where how much of the effects of dietary restriction are sort of mimicked by inhibition of TOR or not. Yeah.
Is it possible to genetically change mTOR late in life in worms, flies, or mice?
Yes, I think the best experiment has been done in mice when rapamycin was added late. But yes, it's we can turn off and turn on things late in life in worms and flies, and I to my knowledge I think it's not been done that well, to see which stage of life is important. But I think the mouse data argues rapamycin works late in life, so this should work. And also, I should add, there's also evidence from, I think, worms and flies, and also in I think other species, that TOR gets activated late in life. So it makes sense. One could argue, therefore, inhibition of TOR maybe in later part of life might be a beneficial thing.
You talked a little bit about rapamycin. Does rapamycin work better than genetic inhibition of mTOR? Do you know something about that?
Yeah, I think it depends on the system. I mean, rapamycin has been a great tool, but really is made against the human mTOR, right? So I think. We've done different combinations. I mean, this is a longer answer, but genetic inhibition in model organisms gives us, more opportunity to explore different questions. So, for example, we found in a very striking experiment that inhibition of both mTOR pathway through inhibition of S6 kinase and insulin signaling simultaneously can extend lifespans by almost 5-fold in C. elegans. Now, with rapamycin, we didn't get similar effects, but my point is rapamycin isn't made to sort of inhibit worm TOR, right? So it may not be working as well, or maybe we didn't get the timing right, like maybe it needs to be switched off during development, right? So we don't know all these questions. So, but I think rapamycin has been a great tool for the field to sort of to manipulate the mTOR pathway, and I think one of the things about these pathways is you don't want to turn them down too much, right? Because the animal with complete loss of mTOR is not viable. These are essential genes for growth and development. I mean, the job of mTOR is to basically turn nutrients into cell mass, and this is an essential gene, but you want to turn it down for some of the benefits. So therefore, the drugs like rapamycin are very useful for that.
If we look at the rapamycin field, what weak points around rapamycin do you see that we need more research on?
I think two things come to mind for me. One of I think the side effects of taking rapamycin, at least very pronounced in in mice, is that there's a level of sort of insulin tolerance or glucose levels go up, right? So I think this needs to be, though this may not be detrimental in some model organisms, it needs to be sort of carefully thought through for humans, and how do you sort of ensure that that's not leading to any other sort of side effects related to insulin resistance. And the other one I think is more important about, I think one of the things, the most fascinating areas I think I find now in aging research is the inter-individual variability. So both with dietary restriction and rapamycin, for example, one of the mistakes that most people make, I think, is say, well, these are anti-aging therapies. And I think this word anti-aging is a problem, because it sort of reflects that it will postpone everything that is related to aging, and that's not true, right? So I want to make this clear that either not, and this has been studied by many people and been argued by many people, it is not a panacea, right? It's not. So one has to carefully think through what health span measures. So to be carefully seen which health span measures would be improved and which ones would not. And I think that's going to be very different across individuals. So some people, for example, with dietary restriction may find big cognition improvement, right? Whereas others might find big metabolic improvement. Some people I feel very active when I am fasting, maybe at the right, I've got it right. But some people feel lethargic. And, so these are things that are very important to sort of work out, which biomarkers, both behavioral and biochemical, are the ones to optimize to benefit, for each health span measure, what you're looking at, and sort of aging of different tissues. Because also one of the side effects of both fasting and rapamycin is sort of wound healing, wound repair can be compromised, right? So you want to be careful when you're doing this, and what times would be safer. Yeah.
Is this something that the Buck Institute is looking at regarding how to individualize the different interventions?
Yeah, I think this is a growing area. We recently just had a conference on role of genetic variants in sort of aging, and I think this area needs more impetus, because I think we kind of think not only we say anti-aging, but we have this idea that these things work for across species and for everyone, and I think that may not be true. That's not going to be true. So we need to understand this at the level of the genotypes as well. So for example, I'm imagining a day could come where, and we've written a very nice sort of a comprehensive review on this, where you would have biomarkers, and you would also know the genotypes, right? And so you could say, okay, if you have this X genotype and these biomarkers, it might be safer for you to do this intervention, or really is going to be much more beneficial for you to do this intervention versus another intervention. And sometimes also the other way around as well. There can be too much of a good thing can be bad for you. So if you're already not really studies show that the longest lived species of flies, they don't benefit more from dietary restriction or possibly rapamycin, right? So people who are already well optimized, they may not get benefits, and sometimes they are the most willing to try more things, and it could end up being detrimental. Yeah.
And this goes a little bit to the next question. If we go to the personal things, do you take rapamycin?
Yes, I have started taking rapamycin for the last year or so. And I think I want to just say it's low dose rapamycin, and then the idea based on a lot of the evidence is low dose rapamycin could be a preventative for many things. So I take a few other supplements as well, and also do intermittent fasting. And the idea is, well, but I have to say I am someone who loves food and overeats, and if I don't do some of these things, I can put on weight quickly. So for me, I think some of these things work well, and it just helps me be more alert, and so I sort of also try to monitor day-to-day differences, and I try not to overdo things in the sense that if I'm exercising those days, I will not try to take rapamycin or some other supplements. Yeah.
You said you take a low dose. What dose protocol are you currently on?
I'm taking 1 milligram per day I'll take it for 2 weeks on and then 2 weeks off. Yeah. And I think this, the lower you can get away with, the better it is. So I try to be in that direction. Yeah.
And if I understand, you take 1 milligram daily for 14 days,
and then I cycle off for 2 to 3 weeks. Yeah.
Okay. What is the benefit in using that dose regimen instead of once weekly dosing?
I mean, I think for me, there are two reasons behind doing intermittently. One, some studies show that intermittent dose is sufficient to give benefits in terms of even extended lifespan. And the second is, with all these things, you want to be on the lower dose if possible, so that you avoid the potential side effects that are there. And the other reason I'm keen in sort of taking rapamycin is, I think low dose, like I said, it could be preventative for many age-related ailments, and including sort of cancer. I mean, 40% of people have a cancer and 20% die of it. Just having low dose of this, if it can help you avoid a cancer, that's another reason to sort of take it. Yeah.
I think the most common dose protocol is around 4 to 6 milligrams once weekly. So it's interesting that you take it daily and not once weekly.
I think a lot of this needs to be worked out in humans. We've not done. I mean, I'm also going a lot more by the mouse data, and there this sort of regimen works. No, I think we need to find out more. I mean, a lot of it is guesswork, I should say, and sort of personal preference on sort of not going overboard. And like I said, I take other supplements as well, so I don't want to overdo. I mean, and I'm also very interested in seeing which things would combine well with rapamycin to extend lifespan further. And like I said, our experiment with insulin inhibition of both insulin and mTOR pathways in C. elegans, which extended lifespan by almost 4 or 5-fold, gives me some hope now that we need to try this in mammalian system, and we're trying to do this in mice right now. And one of the ways we are inhibiting insulin, sort of improving insulin sensitivity, is through this intervention we use called GLYLO, glycation lowering compounds, that we have sort of uncovered. And like I said, so that's one way we're trying to see if these two combined would work. And some data which supports this is, metformin or Acarbose also works well on top of rapamycin. So this fits well with this idea that rapamycin might be increasing your glucose, and that might, if you lower that might be beneficial, especially in humans. But we need to test to see this would be good in humans or not. But I think that's sort of the theory behind this. Yeah.
Have you seen any side effects or felt any side effects of rapamycin?
No, I have not felt any side effects. And one of the things I think I felt an improvement was in my gut. And it's one of the first places rapamycin is supposed to sort of help and work. So, and I could feel that, yeah. And so does intermittent fasting, really help with the sort of insulin sensitivity and also the responses in the gut. Yeah.
Yeah. And have you done any specific blood tests to track if things go in the right direction? Or
I think do my regular tests, and I think sugar is what I pay more attention to right now. I think I'm waiting to see better biomarkers out there. We just developed a biomarker for using retinal scans. I'd be very interested in doing that as this is developed, where you can use the retinal scan to predict the age and also sort of the health of the eye. I'm interested in sort of biomarkers that can give me an idea of the tissue sort of homeostasis. And so I think the general blood work and sort of those kind of, and also sort of just, a lot of tests I think you can do at home, just maintaining, looking at your sort of physical activity responses, for example, how much you can lift, how much, your grip strength and all that, your walking speed, all that. I think they're great biomarkers, and those would be, I track sort of those, yeah. So my stamina and yeah.
And if we look at the risk and benefit calculation of rapamycin, it's still quite early days in research when it comes to humans. Why did you start taking rapamycin and not, for example, another longevity intervention like let's say stem cell therapy or something else?
Yeah, well, I think being close to the research makes a big difference. And like I said, rapamycin and GLYLO and fasting, they are more preventative measures, right? So I think as I get older, maybe if things fall apart, we can talk more about like organismal, to a sort of renewal of other organs. But right now, I think what I think more of is like how to ensure sort of that you can continue to do well for a longer period of time. And so I think everyone has different goals when they're doing this. My goal is more to think of prevention and like, kind of how do you ensure that, everything is fine till 80, 90. And I think I sort of more worked on sort of that basis. Yeah. And your question, I think a lot of the things, I guess a lot of things before they become, well, like let's say they could get clearance to be used, and there's going to be a question about whether, when the time will come for longevity drugs to be ever cleared by FDA. They're not diseases, but there's very little, unfortunately, the system we are in, there's very little on encouraging preventative measures in sort of the healthcare, right? And I think, unfortunately, that the onus comes on the individual and to make that decision, rightly or wrongly. And I don't know. I think everyone is okay to do the way they want, but I think following the research and the sort of seeing that the low dose may not have side effects sort of encourages me to sort of take things like GLYLO and Rapamycin and sort of fasting as well. Yeah.
And you mentioned a little bit about the next thing is that what other longevity interventions can be used instead of rapamycin. You talked, for example, about fasting, dietary restriction, and things like that. Are they additive to rapamycin, or how do you see it?
Yeah, I think this is it's a very tough question, and like I said, it's going to vary a lot across individuals. The only one I can say with a little bit of confidence, maybe additive to rapamycin, are things that would lower, improve insulin sensitivity. And I want to just say, there's exercise, there's sleep, and I believe that you've got to take a healthier lifestyle. I also largely on a plant-based diet, and also I think one of the things I'm becoming more sort of aware of, and I think is very important, is sort of your mental state. I think you can do all these things unless you're in the right state of mind. So things like mindfulness and staying positive, I think probably can even contribute more years to your life. So I think it's a combination of all these things, and I think if you're feeling a day to day, if your activity seems to be good, is one of the best biomarkers you could use to see if you have the energy, and if you have you're thinking clearly. Those are things that can help you sort of optimize.
Yeah, it's a holistic approach. You mentioned that you practice fasting. What type of fasting do you practice?
Yeah, so I think like a lot of research had been on diet restriction, which had been chronic, but I think I've been really taken with the sort of studies on intermittent fasting, where again, periodic fasting can end up being beneficial. And one of the reasons I think it works, at least for me, is that it's easier for me to tell myself to not eat for a few more hours than, okay, not eat, cut down 40% or 30% of your food, right? Or not enjoy XYZ. But it's easier to kind of say, okay, so I do 16-8. So I try to fast for about 16 hours with water, plenty of water actually. And then I often eat 2 to 3 meals, because I'm not trying to lose weight or stuff. But if you're trying to lose weight, then you could go to 1 or 2 meals a day as well. But otherwise, I try to maintain a sort of normal diet, healthy plant-based diet, essentially in those 8 hours. Yeah.
Have you done any research on fasting and life span extension?
Yeah, so in the lab, when we work on this in flies, we find our chronic diet restriction path sort of protocol is much stronger, definitely. But a lot of the benefits, like we recently did the study on sort of improvement in visual function upon fasting, and both intermittent fasting pathways and sort of diet restriction pathways were both working to sort of improve that. So I think, but I also want to say intermittent fasting, really great work has come from Takahashi's lab and also Dudley Laming's lab, working out that both, combination of intermittent fasting and reduction of calorie, not overeating, can be, the best way to probably optimize lifespan. So, and I think that that's been useful to sort of, that's been great work out there, which is sort of, which can help you guide how to do your fasting. Because one tendency, I think I used to have this earlier, and I've checked on this, become more careful about it. I would do my intermittent fasting, and then I would eat whatever I wanted to, or I would indulge more. But now I've sort of, I also did glucose monitoring, and I realized you would get a big spike of food. It's sometimes, I think the biggest driver still is the quantity of food, not even the type of food, even though we pay a lot of attention to type of food, but we often forget that quantity of food is the biggest driver of for example, changes in sugar. So I've just learned to not overeat, and, like they say, eat 10-20% less than what you want to eat, right, for a meal. Yeah.
One thing that you touched a little bit on was GLYLO. Have you seen that it could have any potential additive effects with the rapamycin?
Right. So let me just go into the genesis of that. So our lab's been, one of the things I've been asking a lot is, why is it that sugar metabolism is damaging, right? So like one of the things we found in fasting was you switch over from using sugar to fat. And one of the questions is, well, why is sugar bad, or is sugar even bad? And one of the things we've become very interested in, and it's a fascinating area of research which has been there for a while, is the accumulation of advanced glycation end products. So the idea is, when every cell from bacteria to us uses sugar, and it's metabolized very similarly, and during that metabolism, you create toxic byproducts. One of them is the key one we study is called methyl glyoxal. Now, this is a precursor for advanced glycation end products, because it attacks lysines, arginines, but also lipid and DNA, right? And so, for example, if you had a toast this morning, making the toast creates about 50 new advanced glycation end products. Just the sugars and the amino acids in bread will react to form the ages, which gives it the nice taste. So we've been working on why, unfortunately, these ages are also made in our body. And so we've been working on ways to lower this. And what we found was a combination of commonly used supplements, nicotinamide, thiamine, pyridoxamine, and also lipoic acid and piperine. That combination worked very well and sort of synergistically to reduce the toxicity due to methyl-glyoxal and advanced glycation end products. So we have a preprint out there. It can lower these ages in the serum of the animal, but some of the benefits are huge, a big improvement on insulin sensitivity, and also lifespan extension when given to the mice later in life. So we gave the mice at 24 months of age, and you still saw like more than a 35-40% extension in lifespan at that point. So like I said, I think based on the other data we think this and rapamycin might work well together, and we are attempting those experiments. But I think a lot needs to be optimized to understand how these interventions might work well together. So, but we're attempting these experiments.
Yeah, because one little bit potential side effect with rapamycin can be that it impacts the glucose tolerance and can create some kind of insulin resistance. But if I understand it correctly, the GLYLO can probably help in balancing that in a good way.
Right. Yeah, it's one of the things. Despite I was pre-diabetic, and despite fasting and all that, I stayed pre-diabetic. And since I started taking GLYLO, I noticed my fasting glucose went from 100 in the over 115 to sort of under 90 now, in the 80s and 90s. So I think, and we're seeing some benefits in other people as well. But we have to do clinical trials to see this, how this would pan out in humans. But and you're right, that's the rationale. It hits different pathways. One, we're trying to sort of reduce the toxicity due to the sugar metabolism, and through mTOR, you could imagine enhancing autophagy and reducing protein synthesis and other ways to sort of extend lifespan. Yeah.
How did you discover GLYLO? Can you talk a little bit about that?
Yeah, we uncovered like I said, a pathway while we were studying worms, where we noticed that there are glyoxylases which detoxify these precursors of ages. So our body has defenses from bacteria to us. They're conserved defenses against these precursors of ages. It shows you how important they are. And so we've done experiments where you knock them out. And so interestingly, it's not essential, but it causes dramatic shortening of lifespan and also neurodegeneration. So we have a very interesting model of neuropathy, diabetic neuropathy in C. elegans, which is sensitive to touch just like people. So we discovered a pathway using that basically activation of NRF2 or SKIN1 and C. elegans. That pathway increases the detoxification of these ages. And this pathway seems to be conserved across species. And through this, we sort of, as we got deeper into the research, we were looking for intervention, sort of asking how to pharmacologically sort of detoxify ages. And that's where we started combining. So we had found through a screen of 1,000 chemicals, about 15 hits, and we started combining them in all different combinations. So we used neurons, we fed them sort of precursors of ages, and then we asked, well, what combinations of compounds would limit this toxicity? So then we uncovered this sort of combination of 5 compounds. We thought 3 or 4 would be enough, but this 5 was the best result, what gave us the best results. So we continued with this, and then we asked all these questions, what would do this do in mice? So that's the recent paper that's sort of out there as a preprint, where we found that it has this improvement in sort of reducing appetite by blocking the effects of ghrelin. So one of the surprising things that came out of this research was that we ended up finding a pathway that is also relevant for appetite suppression. So we found that reducing ages not only improves insulin sensitivity, reduces sort of diabetic pathologies, and also extends lifespan in normal mice and improves glucose tolerance in normal mice and diabetic animals, but also that it reduces appetite, and so which sort of fits well with if you think about why we cook food and the role of advanced glycation in sort of making food appetizing. This is like $1 billion industry. All the foods you see, including toasting of cereals and all that is all done to perfection, so they become more palatable, and it's all utilizing the chemistry around sort of advanced glycation end products. And what we have found is endogenously lowering this will also lower your appetite, but it's not sort of, I mean, I take it, I'm not trying to lose weight, but you can also sort of just maintain your weight on it by just eating normally. Yeah. But it makes it easier to do fasting. So I find that it's easier to cut out like snacks, and I have a sweet tooth. I find that it sort of reduces your need to have sort of more carbs, essentially.
Yeah. The advanced glycation end products, are they connected to the hallmarks of aging somehow, or because why don't people talk more about AGAs?
Thank you. That's a great question, actually. So I think there's quite a few reasons. So they were actually quite in vogue in the 80s and 90s, believe it or not. But a few things happened which I think have made it harder. So number one, they're hard to measure. They're not the best tools to measure advanced glycation end products are mass spec based tools, and only a few labs in the world can do this, right? The antibody based methods are not very specific. So secondly, there were a lot of Tony Cerami sort of pioneered this field, and he had compounds to lower this, but unfortunately, when they were doing human trials, they had toxicity issues, so they had to be discontinued, which I think hampered the field a lot, because when a clinical trial fails in an area, it sort of has an impact. So what we have taken is a different approach. We're saying, okay, let's not take drug-like compounds which might have side effects. Let's take commonly used compounds, and that's the reason we've developed the supplement sort of GLYLO, which is using over-the-counter sort of what are called GRAS compounds that are available over the counter. And that combination, what we try to do is use them to activate the endogenous defenses that are there against ages. Right? And I think that's been the difference. So previously it had been used to, the idea was, well, let's trap the ages using sort of chemicals like aminoguanidine. But now what we're doing is more using compounds that would enhance your endogenous defenses against ages and ask if that would benefit, which is working very well.
Mm-hmm. What can the advanced glycation end products lead to? Is it some certain disease or all?
Yeah. No, that's a great question. So to your question about how it connects with hallmarks of aging, it connects with pretty much a lot of them. Like, for example, it's known to impair mitochondrial function. You can imagine critically impairing protein homeostasis, because glycation would be a post-translation modification that's irreversible, ends up cross-linking proteins, right? So the lysines and arginines get modified and cause cross-links which build up, right? So that's why it's called advanced glycation end product, because it tends to be irreversible. But the precursors you can detoxify them, and that's what we're working at. And then also I think one of the strongest, very known, strongly connected with inflammation. So ages drive inflammation. There's a lot of literature, probably most literature is on that aspect. But the key things also to notice is like skin aging, known to be impacted by sort of a cross-linking and advanced glycation. But I think one of the most striking effects we are finding are probably on vascular aging. Again, the stiffening of the blood vessels, all that is happening due to buildup of advanced glycation end products. And that's why we find with GLYLO, you could improve a pulse wave velocity. So it's one of the places, most robust phenotypes we see in sort of improvement in there. So heart function is improved. But also now we're looking at different tissues, so it can slow ovarian aging, it can slow the age-related eye decline in eye function. So I think we're cataloging the effects, and we're working also on the sort of link of this with neurodegeneration, and we're sort of excited about with our sort of recent data that it can sort of reduce age-related, well, in an Alzheimer's disease model, reduce the memory decline that takes place. And finally, I think the area we're getting into is sort of menopause, where menopause accelerates the number of age-related aging issues, and we're finding GLYLO is very good at sort of reversing those. So the insulin resistance, the body weight increase, the cognition decline, those things seem to be rescued by giving the mice GLYLO even after taking out their ovaries.
Yeah, it feels like there are some overlapping also with the rapamycin effects there. So
absolutely, I think, and that's what, yeah, we need to dissect that. At the surface they're overlapping, but if they're mechanistically working or by different pathways, there may be potential to combine them, and that's what we are trying to look at diligently. Yeah.
If we look at the intervention testing program, have you thought of sending in, for example, a proposal to them to test?
No, absolutely. We just we resubmitted that proposal, and I've talked at length to to get advice from people on how to sort of ask the right questions for them. But yes, I mean we've applied for the ITP because one of the things we want to do is ask what happens if you take this early on and do this long term. So we've done the experiments only late in life, and it's good to get this reproduced in sort of different mouse strains. So right now, four or five other labs are also using GLYLO, which has also helped us because we can see, like Andre Bartke's lab is finding that animals that he uses growth hormone, mice that make more growth hormone as a way to sort of accelerate aging, and we can see dramatic improvements in that mouse strain. With Christian Pike, we're working on models of Alzheimer's disease, and it seems to benefit in models of sort of ApoE4. So we're working with different labs. With Matt Caberland's lab, we were working on this model for, they have a model for mitochondrial defects, and that seems to be also improved by GLYLO. So we're working with different labs and the ITP to sort of to see what else can be done with this. Yeah.
If we look at the ITP, the Acarbose and Rapamycin showed the synergistic effects. Do you think we will see similar or better effects when GLYLO and Rapamycin is combined?
Yeah, no, I'm encouraged by the results with Acarbose and metformin. So I'm hopeful, but I think we need to do it, and I think it's not trivial. Like I said, I think there's a fair amount of optimization that's maybe needed, because you can do too much of a good thing, like I said. So you have to get it right. So I think we need to do careful sort of dose response studies, and sort of work out the timings of the interventions to sort of ask these questions. Yeah, but I think there's a lot of interest obviously to see this work, and I think separately it needs to be done in humans. So I think there's a lot of potential here. Yeah.
Is GLYLO open also for the general public, or is it only for research purposes?
Oh no, yeah. To sort of translate the effects of GLYLO in people, we formed a company. Yeah, full disclosure, and it's sold by the company called Jubify, and it's available on Amazon as well. Yeah.
Okay. And do you take GLYLO?
Yes, I take about 2 pills a day. Yeah.
And how do you combine it with rapamycin? Do you take it the same time or a little bit after? Or
the GLYLO and rapamycin I generally take early in the day, like I said, rapamycin I cycle every 2 weeks on and off. And also sometimes I cycle GLYLO as well. Like if I'm exercising, I don't. I take it before exercise, but not so much after exercise. It does seem to, in mice, also improve the endurance. It's another question we're trying to ask. It improves, I mean, it has several benefits, like it seems to increase ketones in nitric oxide, increase sort of, the idea would be to also increases sort of capillary density and blood flow. So we're asking these questions. So I try to be careful not to overdo it, though. Yeah.
And have you experienced any side effects regarding GLYLO? Or
yeah, I think there's mild sort of irritation sometimes in the stomach, but I generally take it with lots of water and food. Otherwise, because it has a piperine, which is essentially a component of black pepper, and lipoic acid, they can sort of can irritate the gut. But if you take it with food, you don't tend to get that. Yeah. Also, sometimes I think some people have can notice, like, because it's a sensitizer, insulin sensitizer, if if your glucose could fall down, and that may not be great, for some people if your glucose metabolism isn't as well regulated. Yeah.
And you talked a little bit earlier about the benefits that you have experienced with GLYLO, like improved glucose levels. Have you experienced any other benefits with it?
Yeah, I have to say, it's a lot of this ends up being anecdotal and sort of based on what your research is focusing on. So I mean, I definitely see improvements, like I said, in making it easier to fast in the gut, and also I think exercise tolerance, again, something I think about more. That seems to be improved, and I think eye health is something I've been paying more attention to. So I think there seems to be improvement, again, anecdotally in there. But I think I've seen some other people comment about that, like older people who are taking 70s, 80s. I have a friend who very carefully monitors he swims almost a mile every day, and we could see there's a slight 10% improvement in his sort of running time, sort of swimming time. Yeah. So I think I think we need to do a lot of research in this direction, and I'm very interested in partnering with people to do clinical trials, because this is not something we do in the lab, but it's something we now understand there's a need for, and it's sort of an exciting intervention to sort of try. Yeah.
Are there other ways we can reduce advanced glycation end products?
I think fasting is a very good way to sort of also reduce, because way we think about it is first at the source, right? So the more you activate sugar metabolism, the more AGEs are going to be produced by the body. So one of the steps is reducing calories and fasting. So when you're fasting, you switch over to using fat, and that would reduce the levels of AGE production. The second part is the detoxification, and like I said, that's where, so turns out very interestingly, GLYLO reduces both glycolysis. We didn't expect that, but it seems to reduce glycolysis and also sort of reduce detoxification. And some of the things we don't understand biochemically, but how much AGEs do you make when glucose is when glycolytic flux is normal? That can also vary, and I think potentially it could help there as well, but we've not measured these flux rates right now. So then there's a detoxification, and then finally is when you get the AGEs are formed, their clearance as well. So I think there are 4 or 5 different steps, and one can I think fasting is perhaps the best thing other than GLYLO that I would know that would reduce AGEs. But also there are going to be genetic differences across people. So some are maybe better at clearing it, and some are not as good as clearing it. And that's one of the reasons we think that why everybody who gets diabetes doesn't have diabetic complications, right? So there are these people called the people who are studied who have had diabetes for a long time, high sugar for a long time, but they don't get pathologies. Whereas other people can have, even in pre-diabetes, they can have a lot of problems, right? So I think one of the mechanisms we think is potentially the accumulation of advanced glycation end products. More than 80% of people who are pre-diabetic don't know they're pre-diabetic. So one can really sort of interventions like GLYLO can sort of help prevent these complications sort of going sort of out of whack, where sustained sort of hyperglycemia can cause problems and tissue damage in the long term, and one can use preventatives, we think, like this to sort of slow down that damage.
Yeah. And you talked a little bit about that how you prepare food can also create advanced glycation and
yeah, correct. So the other source of aging is from our food. So I mean, barbecuing or making food more well done, like or dry cooking, that's what gives the largest levels of ages. And there's some studies, very compelling studies, Helen Valsara's group has done a lot on this, where they found that, for example, in diabetics, if you eat raw food, that can actually help lower glucose. And there are a few ways that can work. I think number one, you can't eat as many calories if you go on a sort of raw food diet, right? Secondly, I think you're also introducing more advanced glycation end products, and Helen Valsara's group has done these studies, and we are finding the same thing. Just introducing ages into the diet is sufficient to cause, shortening of lifespan, accumulation of it prevents breakdown of fatty acids, and insulin resistance. So AGEs just exogenously are also very damaging. So one has to sort of pay attention to that as well, the foods we're eating. Yeah. So steam cooking is better than sort of dry heat cooking.
Yeah. Do you know if rapamycin reduces advanced glycation end products also, or?
Yeah, we've not looked at that yet. Yeah.
No. What do you think? Do you think it can have any impact?
I mean, from the pathways we understand, I think maybe not. But I think it remains to be seen. I could be surprised. I mean, yeah, I think it's a hard question to sort of answer. Yeah. But like, for example, things that would activate NRF2, I think, are one of the ways to sort of reduce, increase the detoxification is one way to reduce the effects. I mean, you could imagine rapamycin, you could also get an increase, because if there's more glucose in the system, and what we're finding like in mice, that could end up increasing the advanced glycation end product. So I think it needs to be, but it's also doing other things like increasing autophagy, which you could imagine some things could be protective. So I don't know the net result. So that's a harder question to ask, and maybe vary across tissues and species. Yeah.
If we start to go a little bit to the end here, we have got quite many questions from the listeners. So if we take some rapid questions there. It came lots of questions to you, Pankaj, and many was about optimal dose protocol regarding rapamycin for longevity, and if weekly dosing is better than every other.
I think we need to do a lot more studies, and like I said, I think one has to think we have to stop thinking of these as the word like anti-aging. They don't solve all problems. One has to sort of start asking, okay, which health span measure are we talking about? And the effects are going to be different on different health span measures, and different doses are going to work differently on that, right? So one needs to be thinking about that. Secondly, I think there are a lot of inter-individual differences. So I hate to comment, like there's not going to be one size fits all. So I think these things make me reluctant to say, I think we need to do this a lot more carefully and to answer these kind of questions.
Yeah, good answer, because the research is limited today, so it's very, very hard to answer those questions. But another question that came is that how well do you expect that the rapamycin will be translatable to humans? Because it has shown very good result in multiple species.
I think that's a very good question again, but it's very speculative. I think like it's already used as an anti-cancer drug. So I'd expect when we're talking about some of the growth signaling pathways, maybe even as a preventative for cancer, like people are using it maybe for prostate cancer. Those are good clinical trials to to test this out, right? Does it prevent as a preventative, will it prevent cancer incidence overall, right? Low dose rapamycin. So that's one. I think there are impacts on gut health, potentially another one. So I think we need to do clinical trials on these different health span measures to kind of optimize these, ask these questions and optimize, what's best and who needs what, right? I think that's another question. Some people may be totally fit and don't even need it, right? So, yeah.
Do you think there exist any additive effects when combining rapamycin with extended fasting for several days?
Yeah, that's a tough question. I mean, I don't know too much about extended fasting. Like I said, we do more intermittent fasting, and I think that has interesting potential because, like, again, it's in a way to sort of improve insulin sensitivity. Maybe like some of the mouse data was supportive that it could be improving further on that. But I'm hate to say what would happen with extended fasting and rapamycin. It may be too much of a good thing, and but I think that needs to be done. That needs to be tested to see if fasting and rapamycin would be beneficial.
Yeah. And where can people find you online?
Oh, well, our company Juvify, I mean, respond to sort of, I can respond to people there when people have questions, but also I'm on Facebook and Twitter, and people have questions.
Yeah. It has been great to talk to you, Pankaj.
Thank you very much for hosting me. This is a great seminar series. Yeah, I mean, this podcast. Yeah, I've enjoyed listening to your other podcast.
Well, glad to hear. Yeah, take care, my friend.
Thank you. Bye.
Disclaimer. The podcast is for general information and educational purposes only, and is not medical advice for you or others. The use of information and other things linked to the podcast is at the user's own risk. Always consult your physician with anything you do regarding your health or medical conditions.